Urinary system. Cells of our body require all sorts of different substances in order to do their jobs, to perform various activity, metabolism. They need glucose for energy, they need vitamins, they need proteins, amino acids, they need sodium and calcium for maintaining membrane potentials. So they also require proper internal environment, water levels, the pH levels, in order to optimally perform all those functions. And the activity of cells produces byproducts of metabolism that they don't need anymore and can't use, can't reuse. They might be toxic. So things like creatinine, ammonia, urea, potassium, those are substances that are either useless or excessive. We have too much of them, or toxic. And so all those substances usually equalize between different water compartments. Remember, water compartments are intercellular, inside of the cells, interstitial, that's between the cells, inside of the lymph system, and inside of the blood vessels, the cardiovascular system. So water and all those substances are going to equalize. They're going to go down the concentration gradients and make sure that there's an equal concentration in each one of them in each one of those compartments. Urinary system serves a function to conserve the useful substances so we don't lose them, and to get rid of all those useless and excessive and toxic substances. They're filtering the blood compartment. So when the urinary system filters the blood compartment, it's going to remove, let's say, toxic substance out of the blood, and then those toxic substances, in order to equalize between the four uh, be, be, between the four water compartments, are going to move out of the cell into the interstitial, out of the interstitial into lymph, out of the lymph into the blood, and then go through the kidney again. And that way we can get rid of pretty much all of the toxic, useless, and excessive substances. The urinary system also reabsorbs all the useful substances. Reabsorbing means taking them out of the kidney, out of the urine, back into the bloodstream. And they secrete the useless, excessive, toxic substances out of the body by moving them out of the blood into the urine and out of the body. So by doing this, urinary system ensures the proper internal environment. It can control the water volume, the ionic balance, the pH. Urinary system filters vast amounts of blood each day. About a quarter of the blood at any given time is going through the kidney. Um, so it's taking in a large amount of liquid, a large amount of water every day, but conserves that water and only secretes the unwanted substances. And those unwanted substances are highly concentrated, and they're suspended in a relatively small amount of liquid, which we call the urine. The functional unit of the urinary system, the business end of the urinary system, where the blood and urine meet and exchange all those substances. Good substances go back from urine into the blood, the bad substances go from blood to the urine. That is called the nephron. A nephron is a tube. A nephron is a tube that is right next to the capillary inside of a kidney. Uh, a few million nephrons that together build two kidneys. The two kidneys are the main organs of the urinary system. Uh, the urine that's formed in the kidneys and then transported, temporarily stored, and then injected from the body by a series of tubes, the ureters from the kidneys to the urinary bladder, where it's temporarily stored, and then through the urethra out of the body. Urine is a clear, kind of yellowish liquid. It is odorless. And why do restrooms stink? Because bacteria metabolize urine and produce urea, ammonia, all sorts of things that might, ammonia is going to be smelly. Specific gravity of urine is slightly above that of water. pH, osmolarity, they can vary widely depending on the water balance, the foods you had, things like that. The color of the urine can change depending on what you ate. Uh, so, for instance, asparagus can turn your, not in everybody, it can turn in many people urine into green or even blue. Garlic is also going to add some smell, interesting odors to the urine. There should be no blood or no red blood cells in the urine. There should be no glucose in the urine. There should be no or very few white blood cells in the urine. There should be essentially no protein or very little protein in the urine. Normal daily volume of excreted urine is between 3/4 of a liter and 2 liters, 750 to 2000 milliliters. If you urinate a greater quantity than 2.5 liters, this is considered polyuria. That can be caused by diabetes mellitus, by diabetes insipidus, by excessive use of caffeine or alcohol, some kidney diseases, excessive water intake, you drink too much, some drugs can cause polyuria. Volume smaller than half a liter is called oliguria, and it can be caused by low blood volume, so hypovolemia or dehydration, which can be caused by bleeding or diarrhea or vomiting, or it can be due to kidney diseases or obstructions like enlarged prostate, which is squeezing onto the urethra and not allowing enough urine out. Daily volume of urine that's smaller than 50 milliliters a day, that's really, really small, it's called anuria. It's essentially no urine. And it can be caused by various kinds of kidney failure, kidney stones, very enlarged prostate that is completely squished the urethra. Um, so that's a serious problem. Urine is excreted via the same openings as the reproductive tract. Reproductive tracts differ between men and women, between males and females of all the mammals, and thus the urethra is differing between the two sexes. In women, urethra is short, only around 4 centimeters, and ends in the vagina. In men, urethra passes through the prostate and other male secondary glands, and those glands are helping clean and sterilize the inside of the urethra, and then it goes through this corpus spongiosum of the penis. So the whole thing, the urethra of a man, is like 5 times longer than that of a woman, which means it's much easier for infections like bacteria to travel up the little, 4 centimeters of the female urethra than all of that, cleaned and sterilized urethra 5 times longer in the man, which is why women are much more susceptible to urinary tract infections. Urethra takes the urine out of the body uh, by taking it out of the urinary bladder. The beginning of the urethra, just as it exits the urinary bladder, has two sphincters. The internal sphincter is smooth muscle, no voluntary control, no conscious control. External sphincter is skeletal muscle, thus it is under conscious control. So that's what you squeeze while you're trying to find a bathroom and run to it when you really, 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 really need to go. Uh, that reminds you probably of the two anal sphincters, right? The internal and external anal sphincters are organized the same way. Internal is smooth muscle, not under voluntary control. External is skeletal muscle under voluntary control. All right, urinary bladder is a sac. It's composed of muscle and the transitional epithelium. You remember it from the beginning of the year, transitional epithelium. So when the bladder is empty, this epithelium is thick, looks like it's columnar, and it's stratified. It's thick. Um, as it gets filled with urine, little by little, it gets fuller and fuller, and that urine is stretching the wall of the urinary bladder. The cells move around each other, and the number of layers gets smaller and smaller as the wall stretches. And the cells, instead of being columnar or cuboidal or round, now look squamous. If it is completely full, like to the bursting, the wall of the urinary bladder can be as thin as a single layer of very thin squamous cells. The detrusor muscle. It is a collection of smooth muscle fibers in various irregular orientations around the entire urinary bladder. Remember the Frank Sterling law from the heart? More the muscle fibers are stretched, greater is the force by which they can contract. And so, more urine there is in the urinary bladder, more the detrusor muscle is going to be stretched, and thus, with a greater force, it can push the urine out of the urinary bladder when the time comes. The nerves that innervate the bladder originate in the spinal cord in the sacral region. Sympathetic nervous system keeps the two sphincters closed and keeps the detrusor muscle relaxed while the bladder is empty. Right? Sympathetic nervous system is kind of controlling the bladder while it's empty. As the urinary bladder fills gradually with urine and the wall stretches, those stretch receptors in the wall of the bladder start stimulating the parasympathetic system. And parasympathetic system will eventually override not just the sympathetic system, but also the conscious control of the external sphincter. There's only so much you can wait before you find a bathroom. And and so parasympathetic system starts the contraction of the detrusor muscle and the beginning of urination or voiding or micturition. So think back to the zebra line example when you're stressed. Sympathetic nervous system and epinephrine are active during stress. And they do what? Inhibit the urinary system, including urination, right? Running away from the lion is not a good time to stop and pee. So remember, it's sympathetic system and epinephrine are going to inhibit the detrusor muscle, and they're going to keep both the internal and the external urinary sphincter closed. The bladder can contain anywhere between zero, after you just went to pee, it's zero, up to about 600 milliliters of urine. 600 milliliters is more than half a liter. That's quite a lot. Um, Once you get to about 150 milliliter, you start feeling it. You're feeling, oh, maybe I should find a bathroom soon. At about 400 milliliters, so that's still less than half a liter, you're running to the bathroom because you're going to pee in your pants if you don't. The voluntary control is now overwhelmed by the involuntary control of the sphincters and the detrusor muscle because the parasympathetic system now overpowers your conscious control of the sphincter. Once urination starts, it cannot stop. Remember, the remodeling of the transition epithelium from a single layer squamous back to columnar stratified is a positive feedback loop, which can stop only once the bladder is completely empty again. So, uh, what has been discovered recently that regardless of how much urine there is in the urinary bladder. Remember, when there's more, it's going to stretch the detrusor muscles more, so they're going to have a greater force, Frank Sterling law. If there's less urine, there's going to be less of a force, so the whole process is going to be weaker and slower. Doesn't matter how big is the animal, from mice to elephants. There's a rule. The act of urination lasts about 21 seconds. The whole thing, from the beginning to the end. Once you start peeing, you can't stop, and it lasts about 21 seconds, and that is not dependent on the volume of the urine, right? The little mouse is going to have a milliliter of urine. The elephant is going to have liters, and we are going to have about half a liter of urine at most. So does it really matter the size of the animal? Doesn't really matter the amount of liquid in the urine. Doesn't matter the size of the urinary bladder. Urination lasts about 21 seconds, 20 to 22. Okay. Ureters. Now we're going back up from the urinary bladder up to the kidneys. Ureters connect the kidneys to the urinary bladder. They're about 30 centimeters long. That's pretty long. They take the urine from the two kidneys and into the urinary bladder. And they enter the bladder at an angle, and an angle acts as a valve, preventing the contraction of the bladder to send the urine back up to the kidneys. So the urine drips into the urinary bladder, and as the urinary bladder fills and expands, it closes that valve just mechanically. The wall is expanding. This is the ureter. Nicely, it's empty. It nicely goes in, sip, sip, sips. As it expands, it's making this angle sharper and sharper. And once it's completely full, it closes it off. There's no more urine able to come in from the ureters. So it's a completely mechanical valve. There's no flap inside. It is a mechanical way of stopping. It's like making a kink in a hose. That's what the urinary bladder does to that very end of the ureter. Um, and so the urine cannot go back up from the urinary bladder up to the kidneys. Ureters also have transitional epithelium, which is surrounded by thick layers of both longitudinal and circular smooth muscles that are moving the urine through the ureters via peristalsis from the kidneys to the urinary bladder. Now, the kidneys themselves, paired organs, there's two of them, one on the left, one on the right. Uh, they are kidney shaped. Well, the beans are kidney shaped, and the kidneys are bean shaped, whatever way you want. You know how they look like. In some animals, one of the kidneys, like the right kidney of the pig, are kind of more triangular, but let's say it's bean shaped, just like the beans are kidney shaped. That's why we call them kidney beans. Kidneys are very sensitive. This is not where you want to get kicked, right? It hurts. It's very easy to damage or rupture a kidney. So they're vulnerable to trauma. So they have to be protected. And they're not really attached by any kind of ligaments or anything like that to anywhere. They're outside of the peritoneum. So they're an extraperitoneal organ, and so they're on the backside behind the intestines, kind of leaning against the back wall of the abdominal cavity. So they're protected by what? They have a thick fibrous capsule, and that is surrounded by very thick renal fat pad. They're floating in large quantities of fat. And then there's the peritoneum in front, and the back wall of the muscles of the back, and the last two ribs in the back. All those structures together kind of keep kidneys firmly in place, anchored against the back wall of the abdominal cavity outside of the peritoneum, and kind of protected from injury to some extent. So if you look at a kidney, and there's this nice, smooth, beautiful, shiny capsule, which is thick and fibrous on the outside. If you remove the capsule and look underneath, what is underneath is the renal cortex. Uh, Renal cortex is where you find the renal corpuscles. And underneath the cortex is the renal medulla. Medulla is made up of several renal pyramids. And between the pyramids are renal columns. That's connective tissue, the extension of the cortex in between the pyramids. This is where interlobular arteries and veins pass. Uh, Pyramids and their columns together form renal lobes. And each kidney is going to have several lobes, depending on the species. But we have maybe a dozen lobes, or even fewer. So each pyramid is formed of collecting ducts, some loops of Henle, capillaries, and each pyramid ends with the renal papilla, where all the collecting ducts end. They all fuse into the renal papilla. So the urine from all the collecting ducts flows from the papilla into the renal calyx. So each pyramid is going to have a papilla, which is the end of all these collecting ducts, and the calyx, which is a little space. So papilla is in the space, papilla is in the calyx. So all the calyces from all the pyramids together form the renal pelvis. That's the hole. That's the uh, that's the cavity inside of the kidney. You know how kidney goes like this. It's like kind of the middle part here is where there's a hole where the urine collects, and then from there goes into the ureters out. Renal hilum is that place. So this is the place where renal artery comes in, renal veins goes out. This is where the lymphatic vessels go in and out. This is where the nerves go in and out. This is where the ureters go out. So hilum is like one place. You know, if you have a kidney bean, there's going to be that one white dot, right? That's the place where renal hilum is located. Everything goes in and out there. All right, now this is what you can see with the naked eye, and we dissected the kidney in the lab, so you saw it, and on the models as well. Now we are going microscopic, what you cannot see with the naked eye, and we are going to look at the nephron. Nephron is the functional unit of the kidney. Each kidney has about 1.3 million nephrons, and each nephron is a tubule. It's a urinary tubule that's filled with urine, and that tubule is surrounded by capillaries filled with blood, and all sorts of exchanges are happening between urine and the blood, the urine in the nephron and blood in the capillaries surrounding the nephron. The nephron starts with the renal corpuscle, so that's where the tube is a little bit bigger, and it's like a sphere, like a little ball. And that ball, that corpuscle, is called Bowman's capsule. The Bowman's capsule is the beginning of the urinary tubule, and so then it narrows down into the tubule. Tubule goes elsewhere. Inside of the Bowman's capsule is the glomerulus. The glomerulus is a bundle of capillaries, and uh, so it's a dense capillary bed that's contained completely inside of the Bowman's capsule. This is where filtration occurs. This is where blood gets filtered into primary urine, and then everything else happens in the rest of the urinary tubule. What is the rest of the urinary tubule? First coming out of the Bowman's capsule is the proximal convoluted tubule. So you have Bowman's capsule, and then you have a very convoluted first part of the tubule, which then continues into the loop of Henle, dips down and up. It has a descending part of the loop and the ascending part of the loop of Henle, and then continues as the distal convoluted tube, a very convoluted part, right, tubule, and then that at the end joins one of the collecting ducts. Multiple nephrons are going to feed into a single collecting duct. And remember, collecting ducts then all join together into the papilla, which drains into the calyx. All the calyxes make the pelvis. Pelvis drains urine into the ureter, ureter into urinary bladder into urethra, and out of the body. Okay, glomerulus is interesting. So that's a capillary bed, right? A lot of capillaries that are inside of that Bowman's capsule. Where does the blood come from? From the afferent arteriole. Brings blood into the glomerulus, breaks down into lots and lots of little capillaries, and then those capillaries fuse and form an efferent arteriole that takes the blood out of the glomerulus. The afferent brings it in, breaks into, branches into a lot of capillaries, and those capillaries all fuse into efferent arteriole. Afferent comes in, efferent comes out of the glomerulus. Then the efferent arteriole branches into a secondary capillary bed. That's a capillary bed that surrounds the rest of the urinary tubule. So you have a vessel, an arteriole, right? You have capillary bed, then have a single vessel leading to another capillary bed. So that is familiar to you, right? That's a portal system. This is called the renal portal system. So when we talk about hepatic portal system or the hypothalamo-hypophyseal portal system, that's one. Here you have 2.6 million renal portal systems. Each glomerulus has its own. So the second capillary bed consists of two sets of peritubular capillaries. Those are capillary beds that are surrounding the proximal and the distal convoluted tubule. They're following those convoluted tubes around. And we have vasa recta, which means what? Means straight tubes, straight vessels. And they surround the loop of Henle. Remember how loop of Henle goes down the descending loop and then up the ascending loop. Well, vasa recta are doing the same thing, but the opposite. The descending loop of the vasa recta goes next to the ascending loop of the loop of Henle. The ascending loop of the vasa recta goes next to the descending loop of the loop of Henle. And so you can already see two tubes transporting liquid in opposite directions right next to each other. There's going to be a countercurrent system there. Okay, cortical nephrons are those that have their glomeruli close to the surface of the renal cortex, right underneath the capsule. They have shorter loops of Henle, and those loops of Henle usually don't even get to the medulla. About 15% of the nephrons are juxtamedullary nephrons. They have glomeruli, which are right there on the border. They're in the cortex, but right next to the medulla. So they're in the cortex, but right on this kind of surface on the other side of the medulla. And their loops of Henle are dipping into the medulla, and they're much longer loops of Henle. Um, so the blood and urine flow side by side in a nephron. Capillaries are surrounding the urinary tubule. And so three processes can occur. First is filtration. Filtration means size dependent filtration of blood into urine, like through a sieve. This is happening only in the glomerulus, only inside of the Bowman's capsule. So pretty much all of blood plasma is going from the capillaries into the Bowman's capsule, forming the primary urine. There are red blood cells, white blood cells, medium and large proteins that cannot go through. Pretty much everything else can. So the blood plasma and primary urine are about the same composition. That is filtration, just filters big things like cells and large proteins. Everything else gets filtered out, everything else goes out. Then in the rest of the tubule, the proximal and distal convoluted tubule, the loops of Henle, and even the collecting ducts, we have two other processes, either secretion or reabsorption. Secretion means that there's additional movement of substances from blood to urine. So that usually happens with the things we don't want, the bad things, the toxic things, the extra things. So filtration is going to, there's going to be a balance. There's going to be about the same in the blood, remaining in the blood, the same in the urine, but you want more to move from blood to urine in order to get rid of it. So there's going to be secretion, active secretion, often using energy to push more of that bad stuff from the blood into the urine and out of the body. The opposite thing is reabsorption. That's the additional return of substances from urine to blood. So once it, kind of blindly just filtered from blood to urine, you have all the good stuff in that primary urine. You want to return all of that into the circulation. You want to keep it. So things like glucose get completely reabsorbed in the proximal convoluted tubule that doesn't even wait much longer. It all completely gets reabsorbed. So you need to know what those two things are: secretion from blood to urine, because it's the bad things you want to get rid of. Reabsorption is from urine back to blood, because it's the good things that you want to keep. Okay, so filtration moves blood plasma from the capillaries of the glomerulus into the Bowman's capsule, where the primary urine gets formed. So the blood cells, red blood cells, cannot go through. White blood cells essentially cannot, but remember they're kind of amoeboid. Occasionally, get one or two may squeeze through, and it's okay if you find a couple of white blood cells in the urine, but not more. Large sized and medium sized proteins cannot go through. They stay in the blood. They cannot enter the primary urine. Everything else passes into the renal tubules. And initially, composition of the primary urine is like identical to composition of blood plasma. The capillaries of the glomerulus, you might remember, are fenestrated. They have fenestra. They have windows. Remember when you throw a king out of the window, that's defenestration. So they have fenestrated capillaries, which means they are very good at controlling the size of the molecules that can pass through. Very little goes through a continuous capillary. Everything goes through the sinusoid capillary. But fenestrated are very precise about the size of the things that can go through those little windows. So in the glomerulus, the fenestra allow the passage of molecules that are smaller than about 8 nanometers in diameter. Now, Bowman's capsule itself is made of cells called podocytes. How do you remember podocytes? Podiatrists, they look like feet. And podocytes have finger like or toe like projections that are called pedicels. Pedicure, toes, right? So podocytes have pedicels, feet have toes. That's how you remember that. Go to the podiatrist for feet, you go to pedicure to get your nails on the toes done. So those podocytes are wrapped around the capillaries and they have those pedicels, right? And so the pedicels are going to be sitting right on top of some of the holes, some of the fenestra. Nothing goes through there. But there's some fenestra in between the pedicels. And this is where some things can be filtered through. Those pedicels can slowly move around, uncover some holes, cover other holes. They can get narrower, they can get wider. Hey, some hormones can actually act on that. And so they are to some extent controlling uh, what goes through the fenestra in between the pedicels. The proteins that are surrounding the fenestra, remember that's cell membrane, and cell membrane is mostly fat, but it has proteins floating around. Remember? There are proteins surrounding the fenestra. And those proteins are negatively charged. So it's much harder for another negatively charged molecule to go through that hole. It's going to be repelled by negatively charged proteins. It's much easier to pass through positively charged molecules, right? They're going to be actually attracted to those negatively charged proteins and then slip through. So charge of the molecule is an additional criterion apart from size is what can go through and what cannot go through the fenestra. Um, Mesangial cells inside of the Bowman's capsule are capable of contracting and capable of contracting uh, around those capillaries. And thus, when they contract, they reduce the filtration rate. They reduce the total volume of blood that is going out of those capillaries into the Bowman's capsule. By doing this, by contracting, they also make it harder for some molecules to pass through. It's actually shrinking the fenestra, right? It's making it harder for some things that can kind of slip through. Once mesangial cells contract, those kind of iffy edge case kind of molecules cannot pass through. So there's a little bit of additional control there by mesangial cells that are susceptible to some of the hormones. Blood pressure in the glomerular capillaries is high. It's very high. Remember that the osmotic pressure and the hydrostatic pressure in the Bowman's capsule are essentially zero. There's nothing happening there. Osmotic pressure of blood is high because there's all these proteins, right? The big proteins are staying in the blood, and the small proteins only can go out. So there's big proteins making big osmotic pressure compared to the urine. So they're going to want to draw water back into the capillaries. So you have to overcome that force by having a large hydrostatic pressure in those capillaries so they can push the water out. So it's really hard. So the net filtration pressure in these capillaries is about 10 millimeters mercury. That's pretty high. How is this accomplished? How is this done? Vasodilating the afferent arterial that's bringing a lot more blood into the glomerulus, and vasoconstricting the efferent arterial that is taking the blood out. So a lot more blood comes in, a lot less blood can come out. That increases the pressure inside of the glomerular capillaries, pushing the liquid out of them through the fenestra into the Bowman's capsule, into the urine. Um, Reabsorption and secretion use several different mechanisms. Remember, they're happening after the urine has left the Bowman's capsule in various different parts of the urinary tubule. And there are different mechanisms by which reabsorption can happen and secretion can happen. Diffusion, passive movement of molecules down their concentration gradient, does not require energy, does not require any pores, it goes right through the membranes, no big deal. Facilitated diffusion is a movement of molecules down their concentration gradient, does not require energy, but requires pores, requires specific pores or channels or receptors, transporters in order to pass through the membrane. So it's facilitated diffusion. There are several different types of facilitated diffusion. There's the simplest. Symport is a type of facilitated diffusion in which two different substances use the same pore or the same receptor, same channel, and they move down their concentration gradients. It doesn't require any energy, but they're going in the same direction. You might have, sodium and chloride going all in the same direction down their concentration gradients. That's called symport. Antiport, type of facilitated diffusion, two different substances using the same pores or channels or receptors. They're both going down their concentration gradients, but those concentration gradients are in the opposite direction. So they're using the same pore to go the opposite ways. Osmosis is the passage of water from areas of lower to areas of higher osmolality, really down its own concentration gradient. If there's like more sodium on the other side, that means there's less water. Water is going to go from where there's more water to where there's less water, right? So that is osmosis. Active transport requires energy. That's a movement of substance against their concentration gradients. So you use transporter molecules that require energy. Cuboidal cells of the renal tubules have many mitochondria to break down glucose and form ATP to energize those transporter molecules that are transporting substances against their concentration gradients. As the active transport or primary active transport. The secondary active transport is the movement of substances down their concentration gradients. So it's down, it's not the opposite. But that gradient is built first by pumps that use energy, like a sodium potassium pump. All right, so primary urine goes from Bowman's capsule into the proximal convoluted tubule. And so a majority of reabsorption and secretion happens there in the proximal convoluted tubule. And the wall of the tubule is made of cuboidal cells, and they have a lot of microvilli, which increase the surface area of the cells. That allows for a large number of pores, a large number of transport molecules to be embedded in all those membranes, just like the alveoli of the lung or the intestinal cells in the intestine. In the kidney, it is the surface that matters. More surface you have, more molecules can pass through per unit of time. So it's not the volume, it's not the mass, it is the surface of the urinary tubules that determines how much urine can be filtered, reabsorbed, and secreted. Most of the water, about three quarters, and most of the sodium, about three quarters, two thirds, about two thirds of water, about two thirds of sodium, is reabsorbed in the proximal convoluted tubule. 100% of glucose, 100% of amino acids, 100% of vitamins are reabsorbed in the proximal convoluted tubule. There shouldn't be any beyond there. All of creatinine is secreted here, 100% of it. Sodium is actively pumped out of the proximal convoluted tubule into the interstitial space, and then it diffuses simply into the capillaries. Many substances symport with sodium, going the same direction. Glucose, amino acids, calcium, chlorine, phosphate. Uh, they use the same pores, the same holes, following sodium. Water follows sodium passively by osmosis. So sodium goes out, water follows in order to equilibrate, to make isotonic fluid environment in both sides of the membrane. Um, Proteins are broken down everywhere in the body and they form ammonia, and ammonia is toxic. A liver converts ammonia into urea, which is not toxic. And we can't use urea for anything, and so it's mostly secreted out. The active pumping of urea is happening much later in the urinary tube. It is actually pumped by the collecting ducts. So all the way at the end, collecting ducts actively pump urea. And as the collecting duct is going, moving urine down itself, more and more urea gets pumped. So if you look at the liquid right outside of the collecting duct, there's a little bit of urea up top, and more, greater and greater concentration of the urea is right at the bottom, at the tip of the collecting duct, which means that there's a gradient of urea concentration right along the loop of Henle. Right, there's a lot less urea, so lot lot less osmolality at the top of the uh, loop of Henle than it's at the bottom of the loop of Henle. And what is urea going to do? Is going to draw water out of the loop of Henle. Loop of Henle is essential for the absorption of water. And how do we learn that? It's kind of hard to study things, microscopic and whatnot, until mid 20th century. How do we figure out what loops of Henle do? By doing comparative anatomy. We compared kidneys of many, many, many different organisms, and we discovered that marine mammals have very short loops of Henle. They live surrounded by water. They drink their water, and plenty. They don't have to save. They don't have to conserve water. Desert mammals, they have hugely long loops of Henle. Very, very long. Various mammals that live in deserts, like kangaroo mice and those kinds of things, have very, very long loops of Henle. So from there we deduce the loop of Henle has something to do with conservation of water, and then later on, kind of figure it out. And there are some links in the additional readings about how we figured out what loop Henle does. The vasa recta, remember, descend along the ascending loop of Henle and ascend along the descending loop of Henle, thus forming a countercurrent multiplier. Actually, two of them. One is next to the descending loop of Henle, the urine is going down, the blood is going up, and the other one next to the ascending loop of Henle, where urine is going up and blood is going down. So we have two countercurrent exchangers, and they're different. And we're going to get into that right now, why they're different. So secretion of urea by the collecting duct produces high osmolar solution in the interstitial space around the loops, greatest concentration being at the tip of the loop. So the water wants to come out. So there's a gradient of urea concentration, which is going to drag water out. There's other things happening here. The descending loop is built of thin squamous cells. Remember the proximal convoluted loop with cuboidal cells with lots of villi? These are thin squamous cells. They make diffusion and osmosis easy, right? The cells have many aquaporins. What are aquaporins? Aquaporins are water channels, channels through which water goes through, and they're always there. They're always open. The proximal uh, proximal convoluted loop and the descending loop of Henle have constituted active aquaporins, lots of them, and they're open all the time. So it's very easy for water to go in and out. Those aquaporins are always made, always embedded in the membranes. The ascending loop of Henle, we're back to cuboidal cells, and there are no aquaporins at all, ever. So water cannot move across the membranes. Whatever is in the tubule stays in the tubule. Can't go in, can't go out, which is very, very important. So as the urine goes down the descending loop, it encounters greater and greater concentration of urea in the interstitial fluid, and so greater osmolarity of the blood in the capillary next to it. So the sodium and water flow out into the interstitial fluid and then into the capillary. And some of the urea actually enters the tubule and travels again. At the tip of the loop, osmolality of urine is very high. It's around 1200 milliosmoles per liter. And that's in humans, as you can imagine. It's much greater in desert mice because the loop of Henle is much longer. It's much smaller osmolarity in whales and dolphins, which have a much shorter loop of Henle. So as the urine now goes up the ascending loop of Henle, the sodium is pumped up, out. The chlorine moves passively in between the cells. There are leaky gaps, junctions there. But water cannot leave the tubule, and there are no aquaporins. So sodium and chlorine move out, while water stays in the tubule. So departure of sodium and chloride and several other ions that follow them passively dramatically reduces the osmolality of the urine at the end of the ascending loop, at the end of the ascending loop at the top, once you go back up. Now, remember, vasa recta moves blood down the ascending loop and up the descending loop. The blood in vasa recta flows very, very slowly. So a lot of water is entering in, right? It takes time to equilibrate. Remember what happens with red blood cells if you put them in hypotonic solution. They expand and burst. So if the blood was going too fast, it would not have time to equilibrate and the red blood cells would burst. So the blood in vasa recta moves very, very slowly. So as the blood starts going down the vasa recta along this ascending loop of Henle, the rising loop of Henle, goes down the rising loop of Henle. It takes in a lot of that sodium and chlorine and other ions that came out of the loop of Henle, which increases osmolality in the blood. As it passes the tip and starts climbing up the descending loop, it has high osmolality, which attracts the water to enter it from the descending loop. And this is where there are aquaporins, so water goes in there. So there's greater uh, osmolality in the blood than in the urine, so water goes from the descending loop of Henle into the ascending vasa recta. Uh, so at the end of that passage, before it becomes peritubulary capillary alongside the distal convoluted tube, the entrance of water has reduced its osmolarity back to normal. So you went from 1200 milliosmoles per liter all the way down to the normal 300 milliosmoles per liter. So the urine now enters the distal convoluted tubule. By that time, about 80% of the water has been recovered, returned into the capillaries, into the bloodstream. So additional 10 to 15% will be recovered inside the distal convoluted tubule. Cells are ciliated, they're cuboidal. Aldosterone is the hormone that controls how many sodium potassium ATPase pumps are embedded in the membranes, thus controlling how much sodium gets reabsorbed, and thus how much water follows sodium passively, thus how much water gets reabsorbed out of the distal convoluted tubules back into the circulation. The distal convoluted tubule is also the place where paired hormone has an effect. Remember, paired hormone is activated, is stimulated when there is too little calcium in the organism. So, what is paired hormone going to do? It is going to control the amount of calcium that is excreted. It's going to stimulate the reabsorption of calcium from the distal convoluted tubule back into the circulation. All right, now the urine goes and finally enters the collecting duct. And the collecting duct is the ADH, the antidiuretic hormone or vasopressin, that plugs aquaporin molecules into the membrane. So remember, proximal convoluted tube and descending loop of Henle, they have lots of aquaporins and they're always there. Ascending loop of Henle has no aquaporins at all. Distal convoluted tubule and especially collecting ducts has aquaporins, has genes for aquaporins and makes aquaporins, but it's the ADH, the antidiuretic hormone or vasopressin, that controls how many of those aquaporins actually get embedded into the membrane, allowing the water to pass through or not. So the ADH controls the final volume of urine. Uh, so remember, the deeper down you go, there's greater and greater osmolality of the surrounding space because it's urea being pumped by the duct, as well as sodium being pumped by the duct and by the loop of Henle. So more water will leave the duct as you go down the collecting duct. And so ADH, by putting the aquaporins in the membrane or not, controls the final volume of the urine, which can be anywhere between 0 and 10% of what was initially filtered from the glomerulus. So, total volume of blood that kidneys filter during the day is about 200 liters, but only about 2 liters are excreted out. Remember, 750 to 2,000 milliliters, three-quarter liter to about 2 liters are normal urinary volume per day. So only about 1% actually gets excreted out. And so the collecting ducts by ADH make the final determination of the actual volume of the final urine. Okay, kidneys are also innervated by sympathetic nerves. So at rest, the stimulation is low, afferent arterioles are vasodilated, there's plenty of flow through them into the glomeruli, plenty of blood gets filtered, and then goes into the kind of vasoconstricted efferent arteriole and the rest of the capillary bed around the rest of the tubules. But in times of stress and in times of physical activity, sympathetic nerves are stimulated. They increase the frequency of action potentials, and that vasoconstricts the afferent arterioles. So less blood goes from the bloodstream into the glomerulus. Less blood gets filtrated by the kidney. There's also less pressure in those glomerular capillaries, so less leaks into the urine, and so more of all of that blood is available to the rest of the body, to the muscles. Um, so kidney is one of the most bloodthirsty organs. It takes up a quarter of the blood in circulation is in the kidneys at any given time. So if the blood is needed elsewhere, kidneys can wait, reduce its activity from the filtration to the final urine production. And so epinephrine has the same effect as the sympathetic system. So just think like a zebra. You don't need to pee. You don't need to make urine because you don't know how long you'll be running away from the line. It could be 2 hours. It could be 5 hours. You don't need. You don't have time to stop to pee. You don't not just don't want to stop to pee. You also don't want your kidneys to produce urine. You want that water to go to the circulation to increase your blood volume, that's blood pressure, bringing all the stuff that your muscles need. All right, so remember how afferent arteriole enters the uh, Bowman's capsule and efferent arteriole exits it right at the same spot. Now, interestingly enough, there's a portion of distal convoluted tubule that just happens to pass by these two arterioles. So now you have these three interesting tubes: afferent arteriole, efferent arteriole, and a piece uh, of the distal convoluted tubule right there next to the very entrance into the Bowman's capsule. And some of the cells of their walls, the walls of the afferent arteriole, some of the cells of the walls of the efferent arteriole, some cells between them, That's a mass of cells that are looking different from your regular endothelium. They call what is called the juxtaglomerular apparatus. Just that means apparatus next to the glomerulus. And these cells can measure blood pressure. How? They have a single immobile cilia stuck inside of the arteriole. And it's always under certain pressure of blood pressure passing by. If the blood pressure is lower systemically, that cilia is going to move. It's not going to be pushed as much. So it is that single cilium, but remember they're like 2.6 juxtaglomerular apparatuses, one next to each glomerulus. So there's a lot of them, and they're going to be detecting lower blood pressure. And so when the blood pressure is low, the afferent arteriole is going to vasoconstrict, reduces the amount of blood entering the nephron, leaving more blood in the circulation. Another response of the juxtaglomerular apparatus is that it secretes and releases a hormone called renin. Renin then goes into the circulation and cleaves some amino acids off a molecule that's normally found there, which is called angiotensinogen. Whenever there's a gen in the end, you know it's inactive form. So renin activates in angiotensinogen. It cleaves off like an amino acid off of it, and what you end up having is angiotensinogen turns into angiotensin 1. Angiotensin 1 is now in the circulation. It's still not an active form. Lungs are also feeling the low blood pressure, and lungs release an enzyme called ACE. And so now you have angiotensin 1 in the circulation, you have ACE in the circulation. ACE can act on angiotensin 1. And when ACE, the enzyme, acts on angiotensin 1, it activates it. It turns angiotensin 1 into the active angiotensin 2. Angiotensin 2 drives vasoconstriction of blood vessels around the body. It's mostly arterioles. Remember when we talked about blood vessels, arterioles are the major controllers of blood pressure. And this will bring the blood pressure back up. There are arterioles in the kidneys too, right? The afferent arterioles. They're also going to respond to angiotensin 2, and they are going to vasoconstrict. That further reduces the blood flow into the kidney, right? So through the kidney and into the urine. Thus, there's more blood available for the general circulation. Thus, you increase the blood volume, which increases the blood pressure. And that should be enough for the urinary